Hey AP Bio students, just wanted to check in with you and see how things are going with your studies of Unit 2 and transport. This is the PhET simulation, PHET, that I linked on the week 9 agenda. Hope you'll give it a couple minutes of your time to check out, just kind of look at what you notice going on here. We've got a nerve cell, the inside of which is colored blue, and the membrane of which is colored yellow. And what are some things you notice here in this nerve cell? Yep. Okay, awesome. If you're saying that it's got different membrane channels, that's great. What kinds of molecules are these membrane channels? They're proteins. They have a 3D shape. You got it. So again, the shortcoming of the model is it's 2D. It looks flat, but you know this is a 3D thing. And are they specific? Meaning, do the protein channels only work on one type of ion? Hopefully you can watch it for a minute and generate an idea. It seems like the teal one only allows potassium to travel through it, and the orange one only allows sodium to travel through it. Again, I'm looking over here at the key to know about the shapes and colors of these things. Do they seem one directional? Do they seem like, well, they allow things in, but not out? No, not really. I see two-way travel if you watch it for a little bit longer. So what do you think is dictating the movement of these ions? How do they know whether to move in or out and at what rate? And so another thing you can do is look at like, well, we probably would want to know what else is going on inside and outside of this nerve cell. Are there any concentration gradients? Meaning, are there any differences in the concentrations of chemicals? In this case, sodium and potassium are the only that are shown. Any differences in how crowded the sodium and the potassium are, either inside or outside the cell? And hopefully, it's not quantitative data, I'm not counting them all, but hopefully just by looking, you can say, yeah, there's way more potassium, there's a higher potassium concentration inside the cell, and there's a higher sodium concentration out here, outside the cell. So you would probably predict then that potassium is going to move out of the cell and sodium is going to move into the cell until they reach equal concentrations. And so another thing you can do is click to see the concentrations in millimoles. And hopefully they change a bit as these chemicals diffuse in and out of the cell. The other thing to think about, if it seems like, why aren't the overall concentrations changing, is that there is already a charge inside of this cell. And so if you click the charges, here, I'm gonna take this off so it's not as crowded. If you click the charges arrow, you can see, you can see that the inside of the cell is negatively charged. But why, you might say? Like, it's full of positively charged potassium ions. Why would it have a negative charge? And so they haven't shown in this model some of those really large macromolecules that are making the inside of the cell negatively charged. Can you think of any? DNA is one. Awesome. There's DNA inside of the cell, and that has a negative charge because of all those phosphate groups. Proteins are going to be negatively charged because they have all of those nitrogens and oxygens. So those are inside of the cell and they're lending this negative charge. So one thing where we're headed to start thinking about is not only can you have a chemical gradient, meaning a difference in the concentration of chemicals inside and outside of the cell, you can also have a electricity gradient, meaning an electrical charge difference. So here we have an electro and a chemical gradient, sometimes just referred to as an electrochemical gradient, in the fact that we've got a negative charge building up inside the cell and positive outside, and then we've got differences in our chemicals of sodium and potassium. So this is the foundation of how batteries work. By separating the charges into a positive and a negative compartment, you have the potential to do a lot of work. So that's how, similarly, how our cells have the potential to do work. They can create or maintain concentration gradients of electricity, of chemicals, and we'll even look at pH later, how pH gradients can also be established. And that gives the ability then to conduct or do work. Okay, so preview of where we're headed. All of these are showing facilitated diffusion, right? It's the movement of ions. So we know those don't diffuse, that's not simple diffusion. They're not moving right through the cell membrane. They're moving through carrier proteins or protein channels. And so that is just facilitated diffusion. So to take it a step further today, let's do a little review of active transport as well. So if you go into our class and go to modules, all the way at the end of week 2 is, all the way at the end of unit 2 is week 9. And I'm on the week 9 slides all the way at the end, slide 88. And so in this case, hopefully by the end of this little review video, if you don't feel like you can already, you'll be able to really specifically describe how active transport works. How can we move chemicals against their concentration gradient, meaning from an area of low concentration to high concentration? Also, by what happens by active transport is any kind of movement of really large molecules, things that couldn't fit through a carrier channel protein. So in the beginning, on the PhET simulation, we were looking at how sodium diffuses into cells because it's higher, the concentration's higher outside than in, so sodium comes in, and how potassium concentration is higher inside the cell, so it diffuses out. And so then in order to restore the starting concentrations where you had this difference, it requires work, it requires energy, it requires an actual pump that is then going to kind of reset back to the beginning the way it was when sodium was higher outside the cell and potassium was higher inside the cell. And so the special protein enzyme that does this is called an ATPase, and it is the sodium potassium pump. So when we say it requires energy, we're talking about energy from ATP. And so what actually happens is the ATPase, the sodium potassium pump, is phosphorylated, meaning an inorganic phosphate from ATP attaches to it. And this is going to shift its 3D shape just a bit to allow the molecules then to bind and be released inside of the cell for sodium and back outside of the cell. So here's a little video clip of it from the textbook that's kind of like a very simplified model, but I think it's helpful to start there.
The sodium-potassium pump is an active transport mechanism. 3 protein channel and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
Okay, so if you want to watch that video again, it's linked right here at the top of our slides. And then I also wanted to show you one from the Protein Data Bank. So they're actually using like the 3D shapes, and it helped me visualize like all that we've been studying with proteins and enzymes and molecule, how this works. So if you want to watch the whole thing, I would totally recommend it, but I'm not going to show the whole thing here. If I can.
Neurons in our nervous system use electrical impulses to transmit information. The sodium and potassium pump restores the sodium and potassium gradients, making the cell ready to send another signal. With each pumping cycle, this transport protein moves 2 potassium ions back into the cell and 3 sodium ions out of the cell. Because the pump moves these ions against their concentration gradients, it requires the energy of 1 ATP molecule for each cycle. This molecular machine comprises 3 chains, alpha, beta, and gamma. Within the alpha chain, 3 domains, A, P, and N, form the core engine of the pump. The pumping cycle occurs in several steps. With ATP bound in the N site and the transmembrane region open into the cell, the pump binds 3 sodium ions from the cytoplasm. This causes the N, P, and A sites to shift closer together. With the help of a magnesium cofactor, the P site is phosphorylated and ADP is cleaved off.
Okay, so there you saw the actual ATP adenosine triphosphate, so 3 phosphates attach. You saw the cofactor, the magnesium, help kind of attract it in, and then the bond between the third phosphate and here's the third phosphate and the other 2 was split. So in doing so, in hydrolyzing ATP into ADP, that's going to provide then the impetus to change the shape of this pump so that now it will be more favorable to accepting the sodium ions, 3 of them, from the inside of the cell, where it's going to pump them back out to the outside. And I love how this shows that lipid bilayer that we've been studying.
This leads to a conformational change that exposes the sodium ions to the outside. The sodium ions can then be released, one after another, into the extracellular space. In this ion empty state, the pump allows the entry of 2 potassium ions.
There's the 2 potassium.
This causes dephosphorylation of the pump followed by closing of the extracellular gate and allows the N domain to accept ATP again.
So did you see that one singular phosphate group go off? You'll usually see that abbreviated P with a little I for inorganic, meaning it's no longer attached to carbon-containing molecules. And so then now this whole cycle can repeat. So one of the things, hopefully, if you have the Principles of Life textbook, was to explain what was going on in one cycle. If you're doing the interactive textbook, you had lots of quizzes and stuff on how that works. So please let me know if you have any questions on the sodium potassium pump. It's a great example of active transport. And the other big one that we're going to look at is proton pumps. We'll see them in the next unit more significantly. So last 2 forms of active transport that you need to know are co-transport, meaning multiple molecules are being transported at the same time. So whenever you see that think, together, or same time, like co-pilots working together cooperation, covalent, right? They're sharing valence electrons. So here you've got two different or maybe even more molecules being transported at the same time. I think this one's really cool. So let's see how it works.
Small molecules, such as sugars and amino acids, can be transported up a concentration gradient. The sugar moves via a membrane transport protein from outside of the cell where the sugar concentration is low to the inside of the cell where the sugar concentration is high. The transport of the sugar through a coupled transport protein is driven by the movement of counterions such as sodium ions or protons that are moving down their concentration gradients from a region of high to low concentration.
Okay, so that was fast. Sodium is moving from an area of high concentration. Remember, it's higher on the outside of the cell. And so it diffuses into the cell through facilitated diffusion, just moving through the protein channel. Then the sodium potassium pump restores the higher concentration of sodium outside the cell. But this, I picture this as like riding along with it. So the glucose in this case is kind of riding along with the sodium. The sodium's flowing in and the glucose is like catching a ride through this special coupled protein channel.
Sodium ions and the specific sugar or amino acid simultaneously bind to the same transmembrane protein on the outside of the cell, called a symport. When the counterion is sodium, the low concentration of sodium on the inside of the cell required to transport the sugar is maintained by the sodium-potassium pump, which is powered by ATP. In a related process called countertransport, the inward movement of sodium ions is coupled with the outer movement of another substance such as calcium ions. As in cotransport, the sodium ions and the other substance bind to the same transport protein, called the antiport, but in this case they bind on opposite sides of the membrane and are moved in opposite directions. The low internal sodium ion concentration is maintained by the sodium-potassium pump.
So a similar process now is using a coupled transport protein called an antiport if the two molecules are moving in opposite directions. So if one is moving out of the cell and the other is being moved in, that's active transport and it's using an antiport. If both are moving into the cell or in the same direction, it's called a symport because they're moving together. Okay, so just a couple hopefully helpful visuals on these kind of complex processes. Please let me know if you have any questions on that. Finally, there's bulk transport, which is basically moving large molecules either into the cell, which is called endocytosis, or large molecules out of the cell, which is called exocytosis. And the vehicle for this is a vesicle. So the way this happens, the mechanism for it is through a vesicle. So for endocytosis, something, either food, fluid, or maybe even an antibody, a ligand, some kind of binding molecule will approach the cell membrane, and the cell membrane will fold. Like here's our cell membrane, it will actually fold in and surround the solid, the liquid, or the molecule, and kind of bend in with it and pinch in and bring it in. Now, if it's something to be digested, maybe it's like a lysosome kind of vesicle that has enzymes that'll help break it down. You don't need to know these vocab words, but you may see them. 3 types of endocytosis. If it's a solid that's going to be digested, it's called phagocytosis, phago being the Greek word for eating. So it's going to be like digested. If it's a fluid, penocytosis, meaning it's brought in and then drinking. Peno is the Greek root for drinking. And then receptor-mediated endocytosis means it's not just automatically like, sure, come on in. It's like something has to bind with a receptor on the surface of the cell, and then that unlocks and allows something in. And so this is the case with viruses. It's sort of a mistaken receptor-mediated endocytosis because it's not in the cell's best interest most of the time to allow a virus in. But the virus's protein spikes are similar enough to protein spikes that our cells would normally allow to bind and allow in that it kind of makes a mistake and allows them in. So I thought we could take a look at a couple questions. One from the AP Classroom, the progress check that I thought was tricky about this, and then another just as like a practice before we wrap up. So you know, big applications, I think we talked about briefly about the nervous system and how neurotransmitters can be released by exocytosis of one nerve cell and then brought in by endocytosis by another nerve cell across that synaptic cleft, synaptic gap, whatever you want to call this here. So for one cell, it could, you know, the cell that's pushing the thing out, for cell A, this is exocytosis, right? It's like, bye, exiting. For cell B, the same kind of process is bringing in these neurotransmitters. That's going to be endocytosis. So it's all sort of relative of like which cell you're talking about and what we're referring to. All right, so if you want to pull up your personal progress check, if you haven't finished it yet, question 16 tells us that lysosomes digest food particles brought into a cell by endocytosis. Okay, so let me give you a second just to pull up your personal progress check, or pause and then just go grab it. All right, and so we'll do that one in a second. So we're looking at question 16 from the personal progress check. Okay, so lysosomes digest food particles brought into a cell by endocytosis. Okay, so first things first, I'm already thinking, okay, we're talking about active transport here because we're talking about endocytosis. So I'm already thinking this is active transport. After a vesicle containing food particles fuses with a lysosome, H+ ions or hydrogen ions are transported into the lysosome from the cytosol. Okay, so if we had a picture here, you know, some food particle are these dots, and they're going to be brought into the cell, into a vesicle, and then H+ ions, once that happens, H+ ions are going to be brought into the vesicle, into the lysosome vesicle from the cytosol, from the surrounding fluid. And if you don't remember, we'll review it more in the next unit. H+ ions, this is one definition of an acid. So this equals acid. And we know that acidic enzymes can break down proteins. They can denature proteins. So if this is some kind of food source, these H+ ions are going to help break down the food. This significantly lowers the pH of the lysosome, sure, and activates the enzymes that digest the particles. Okay, which of the following best predicts what will happen to the lysosomal enzymes if the proteins that transport H+ ions from the cytosol into the lysosome are damaged? So the food particles are still being brought in, the vesicle is still forming, but the problem is here. The H+ ions are not being brought into the lysosome. And so what did those do? Well, they helped activate the enzymes that digest the food particles. So before I even look at the answers, my prediction would be this lysosome is not going to be able to digest the food particles because the enzymes that are activated by the H plus ions that come in are not being activated. So like we talked about pepsin breaking down proteins, and it had an inactive precursor named pepsinogen. It's those H plus ions that actually are the message to convert the inactive pepsinogen into the active pepsin. Trypsin has a similar thing going on for those of you that did that one for your Jollicule. So let's take a look at the options. The lysosomal enzymes will not become active since there will be no active transport of H plus ions. Ooh, that sounds really good. Let's keep our eye on that one, but let's also read the others because maybe there's one that's even better. The lysosomal enzymes will not become active, I agree, since H plus ions will diffuse out of the lysosome. No, they never are. This is saying that they're never being brought into the lysosome. They should be transported into it, but if the enzymes that transport it from the cytosol into the lysosome are damaged, then they're never even being brought into here. So it can't be B. Lysosomal enzymes will become active since facilitated diffusion will move H plus ions into the lysosome. The lysosome enzymes will become active since passive diffusion will move H plus ions into the lysosome. Okay, well, we know it's not D because H plus ions are ions, and so they will not passively diffuse. So the question here for you is really, do you think, oh no, it doesn't say that they will not become active. Well, if the proteins that transport them are damaged, then they won't be able to move H plus into the lysosome. So we have to rule out C. Even if it is facilitated diffusion, this is saying that the proteins that allow that are damaged. They won't be able to. So A is the best answer here. Although the reason that I thought this question was tricky, and if you saw me hesitating a bit in answering it, I'm trying to answer it just with the knowledge you have right now, just with the knowledge from this question. And so they haven't necessarily told us that H plus ions are moving against their concentration gradient. And I wouldn't expect you to know that yet. However, there's nothing incorrect about A, and there's no better choice in B, C, or D. So hopefully this one was a struggle, like a little bit of a productive struggle in like wrestling through why is B not the best? Why is C not the best? Why is D? But again, talk to me, let me know if you have questions on that one. Okay, one more to talk about here, and that one is going to involve a contractile vacuole. So we started out, I think, with this unit watching a video of this single-celled eukaryote. So it's not a bacteria, it's a eukaryote. It has a nucleus, it has mitochondria, it has membrane-bound organelles. You can see cilia on the outside of it. It's actually one of the rare single-celled eukaryotes, and it lives in a freshwater environment, which means it's surrounded by nearly 100% water. And so water is constantly diffusing into this paramecium through aquaporins or facilitated diffusion or osmosis, however you want to describe it. And so the paramecium has evolved this contracting vacuole that pushes the water, contracts, and you can see it down here, pushing the water back out, like, okay, that's enough, back to your freshwater environment you go. So it's, watch it contract and then kind of squishes the water back out into the pond. Pretty cool. And so a lot of times you'll see questions about these on the AP And so let's look at one as an example. So here we have the scenario that researchers are going to be looking at how the amount of ATP used by the contractile vacuole, it's got to be active transport to push it back out. It's flowing in by passive transport. It's not taking any energy to flow in, but it is taking energy to pump it back out from an area of lower concentration to pure water with the environment that it lives in. So how does the use of ATP, the use of energy for active transport, change as you increase the salt in the water, as you increase the amount of salinity or salt that's in this paramecium's environment? And so before we look at the question or the answer choices, let's just take a look at the graph and see what we think is happening. So we know that it's active transport to pump the water back out. And that's what the contractile vacuole is doing. So it's requiring ATP. And so as you increase the amount of salt, the amount of salinity in the environment, so going across the x-axis is increasing the amount of salt, then the amount of energy needed is decreasing. So less active transport is happening as the environment is becoming more salty, as the environment is becoming more hypertonic. And so that makes sense, right? Because the difference in tonicity, the difference in the concentration of stuff is becoming less drastic. The concentration gradient is decreasing as you add more stuff to the area outside of the paramecium. So right now it's just pure water at the beginning, but if we start putting solutes into the water, then there's also stuff inside here. Maybe they're closer in concentration and less energy is required because less water is diffusing into it. Okay, so let's see what they want us to do. Of the following, which additional investigation can be used to determine when the cells are in an isotonic solution? Ooh, okay. So in an isotonic solution, we know that the concentration of solute, and if I haven't said it out loud specifically, these brackets mean concentration, that the concentration of solute outside the cell is equal to the concentration of solute inside the cell. That would be isotonic. Iso means same. Tonic is the measure of how much stuff there is, solute. So we looked at this with our potatoes, right? We found on the graph when they were in a solution, or we predicted what concentration of stuff would have been equal to the concentration of stuff inside them. And we found that graph at the point where they had zero change in mass. So this graph would tell us the point at which they are isotonic to their environment would be the point at which no active transport was happening, meaning no water was being pumped out and no net water was diffusing in. So we would be looking for the point at which no ATP is being used for this active transport. And so when do you think that'll be on this graph? I don't think we've hit it yet, right? There's a general trend of decrease. If I extrapolate this out, it's going to be somewhere a bit more salty than we already are. So the investigators, in order to reach that isotonic solution, they're going to have to increase the salt or increase the salinity a bit more in order to get to the point where no ATP is being used by the contractile vacuole. Of course, there's going to be ATP still used by the organism for all of its other processes, but for the pumping, just for the pumping of water out, that won't be happening anymore because it'll be in an isotonic solution. So we can rule out A and B because they talk about decreasing the salinity. And then of our choices, C or D, we want to increase the salinity a little at a time until ATP use reaches a minimum, because that tells us where the contractile vacuole is not having to work anymore. It's not having to pump the water out. All right, thanks everybody who tuned in today just to do a little review of active transport. Please let me know if you have any questions, and I'll look forward to seeing you on hopefully maybe check-ins tomorrow or at class on Thursday. Thanks everyone. Happy November.